Hello everyone, welcome to TryRight. This is Pack 365, myself Srija. So today in this session you are going to learn about the physiology of reproductive system. So what are the functions does male reproductive system do? So HPT acts as nothing but the hormonal regulation. So this is the hormone control system that regulates the testosterone production and sperm formation in the males. So you have to think this as a three-step chain. The first is hypothalamus, which is a boss, and pituitary, which is a manager, and testis, it is a worker. The first part, hypothalamus. So hypothalamus, it's a control center in the brain. So what does it do? It releases a GnRH, which is a gonadotrophin releasing hormone. So GnRH, it is released in the pulses, which is not continuous. So this push pulses, they tell the anterior pituitary to release its hormones. So you have to think this GnRH as a signal that starts the whole process. So the second is anterior pituitary gland. So this is stimulated by GnRH again. So it releases two important hormones. The one is LH, which is a luteinizing hormone. So this travels to the Leydig cells in the testes. It makes them to produce the testosterone. And the next hormone is FSH, the follicle stimulating hormone. It acts on the Sertoli cells. It helps in the sperm formation, the process called as spermatogenesis. So it triggers the production of androgen binding protein, which holds the testosterone inside the testes to support the sperm growth. So the pituitary, it is the manager sending orders to the testes. And the third part is testis, which is a target organ. So there are two important types of cells in this testis. The one is Leydig cells, the other is Sertoli cells. So Leydig cells, they are stimulated by LH. So they produces the testosterone, which is the main male hormone. So what are the functions of testosterone? It helps in sperm production. It maintains the male secondary sexual characteristics like muscles, deep voice and facial hair. It is required for libido, nothing but the sex drive. It supports the growth of the male genital organs. So now the Sertoli cells, which is stimulated by FSH. So they help in the nourishing developing sperm. So they releases the inhibin. So inhibin is nothing but it sends a message back to the pituitary to reduce the FSH hormone, which creates a negative feedback control. So Sertoli cells, they are the nerve cells for sperm. So testosterone, it travels in blood in two forms. One is free testosterone, which is the active form, and the other is bound testosterone, which is bounded to albumin or SHBG. And here there is a picture of the hormonal regulation. And in the step one, the hypothalamus, it releases the GnRH. So this GnRH is released in the pulses, which is important for the normal hormonal function. And next is the step 2, pituitary releases the FSH and LH hormones. And then the step 3, testes, they respond and it releases the Leydig cells as well as the Sertoli cells. And then Leydig cells, they make testosterone, and Sertoli cells, they support the sperm formation, and then it creates a negative feedback, which is a control system. So this diagram shows the feedback loops. Like high testosterone, it stops the hypothalamus and pituitary, it reduces the GnRH, it reduces the LH, it reduces the testosterone production, which keeps the levels normal. Whereas inhibin, it stops the pituitary, like it specifically reduces the FSH. So this keeps the system balanced like a thermostat. So we have discussed this about the Sertoli cells and the negative feedback, right? So testosterone, what does it do? It is a male sex hormone, right? So it inhibits the hypothalamus and pituitary. The testin, the inhibin, it selectively inhibits the FSH. So it acts, this FSH, this acts again on the Sertoli cells. Next is spermatogenesis. So what is spermatogenesis? It means the production of the sperm. Like it happens in the seminiferous tubules of the testis. So it takes about 64 to 74 days for one sperm to fully form. Where it happens inside each testis, there are many seminiferous tubules, which are the tiny coil tubes. So along the walls of these tubes, they are the special cells like the spermatogonia, which are the starting cells, Sertoli cells, which support and nourish the sperm, and Leydig cells which are present outside the tubules, they produces the testosterone. So there are three main phases of the sperm production. The one is spermatocytogenesis, and the second is meiosis, and the third is spermiogenesis. So what is spermatogenesis? Spermatocytogenesis, what does happens here? This is a starting phase. So what happens here? The beginning cell is called as spermatogonia. This is a diploid 2-end cell. So they divide by mitosis. So what does mitosis helps? They keep some cells as stem cells, right? So they convert other into primary spermatocytes. So what is a primary spermatocyte? It is a larger cell that will start the meiosis. So the second phase is meiosis phase. This phase, it reduces the chromosome number from diploid stage to haploid stage. So this is important because each sperm must have 23 chromosomes. So there are two steps within the meiosis. So in the first, the primary spermatocyte, they develop into secondary spermatocyte. So this is the first meiotic division. The chromosome number reduces from diploid to haploid. So now the cell is called as the secondary spermatocyte, which is in the haploid condition. And the second step is this secondary spermatocyte, they convert into spermatids. So this is a second meiotic division. They produce as the spermatids. So spermatids are haploid, they are but still round and immature. And the third phase is spermiogenesis. So this is a maturation of spermatids. So this is not the cell division, it is a cell transformation. So spermatids, they gradually change into mature sperm, which is called as a spermatozoa. So what are the major changes happens in this spermiogenesis? The major changes are formation of the acrosome. Acrosome, it's a cap filled with enzymes. It helps the sperm to penetrate the egg. So the next change is development of flagellum. So this is a long tail that helps to sperm to swim. And the third change is condensation of nucleus. So genetic material becomes tightly packed. So head of the sperm becomes smaller and streamlined. The fourth change is shedding of the excess cytoplasm. So extra material is removed. It helps to form a light, fast-moving sperm cell. So what is the final result? A fully formed spermatozoon with three main parts: head, midpiece and tail. So head contains nucleus plus acrosome. The midpiece, it is packed with the mitochondria, which is the energy for the movement, and tail, it propels the sperm forward. And this is a spermatogenesis diagram here at the right side. So this diagram shows how the sperms are made inside the seminiferous tubules of the testis. So testis, it contains the seminiferous tubules. So seminiferous tubules, this is a place where the sperm develop. And epididymis, this is where the sperm are stored and gain motility. So you can see here the phases. So the first phase is the spermatogonium, which is a diploid stage. It converts into the primary spermatocyte within the mitotic division, and in the meiosis 1, it develops into a secondary spermatocyte, and by the end of the meiosis, it becomes a spermatid. So this spermatid, it converted to the sperm cells, which is in haploid condition. So this is what the diagram shows you. And spermiation. So what is spermiation? It is a final step of the spermatogenesis. So what happens in this phase? Mature sperm are released from the Sertoli cells. So they move into the lumen of the seminiferous tubules. So from there, sperm start traveling toward the epididymis. So what is a maturation and storage of sperm takes place in epididymis. So what is epididymis again? It is a long coil tube sitting on the top of the testis. So what are the main functions? It is a main site for the sperm maturation. The sperm learn how to swim, how to gain fertilizing ability, and the sperm can be stored for several weeks in this epididymis. So immature sperm, they entering the epididymis, they cannot fertilize an egg. So only after spending time in the epididymis do they become capable, they become mature. Unless and until the sperm reaches epididymis, the sperm is still immature. Okay. So next is the vas deferens. So vas deferens is a thick muscular tube that carries the sperm during the ejaculation. So what is the function of vas deferens? Peristaltic contractions, they push sperm forward during the ejaculation. So this can store sperm for 3 to 4 days. It connects epididymis to the ejaculatory duct. So how the semen formation is done? So semen formation process. So it is done by the axillary gland function. So semen is nothing but the sperm plus fluid from the axillary glands. So axillary glands, they produce most of the fluid that protects and nourishes the sperm. A is about the seminal vesicles, which produces 60% of the semen. So these glands are these add the largest amount of fluid to the semen. So what does their fluid contain? Fructose. So fructose, it's a sugar that provides energy for sperm movement, like fuel for the sperm's tail, like flagellum. And prostaglandins, this helps the sperm to move better. They help the female reproductive tract muscles to contract and to pushes the sperm for upward. And fibrinogen. So this helps the semen to coagulate just after the ejaculation. Like this helps the semen to stay inside the female reproductive tract. So later it becomes liquid again to allow this sperm to swim. So seminal vesicle fluid, it is an alkaline which helps to neutralize the acidic environment of the vagina, protecting sperm. So here there is a diagram which shows you the production of the from production to ejaculation, like what is the flow of the sperm. So the first step, the sperms are made in the seminiferous tubules, and in the second, the testosterone and FSH hormone, they help to the sperm to develop. And the third step, the sperm move to the epididymis for the maturation. And the fourth step, the sperm travel again through the vas deferens. So in the fifth step, the seminal vesicles add nutrient-rich fluid to this sperm. And sixth, the prostate gland adds more fluid to the sperm. And the seventh, the semen exits through the urethra during ejaculation. So this is the flow of the sperm. And accessory glands, prostate plus bulbourethral glands. So prostate gland, what is the physiology of prostate gland? It produces 30% of the semen. So the prostate, it adds a milky white fluid to the semen. So why does its fluid contain and why? So enzymes like PSA, prostate specific antigen. So this helps to liquefy the semen after ejaculation. So semen initially thickens, later it becomes thin so that sperm can swim easily. And it contains a citrate, which is a nutrient used by the sperm for energy production. It contains zinc, which helps to stabilize the sperm DNA, and it also protects the sperm from damage. And alkaline pH, it helps to neutralize the acidic vaginal environment. It protects the sperm and improves their survival. So bulbourethral glands. So these are the small glands located below the prostate. So what are the functions of this bulbourethral corpus gland? Mucus secretion. So this is released before the ejaculation. Nothing but it's called as a pre-ejaculate. And it lubricates the urethra so that the sperm can pass smoothly through the urethra. And it also neutralizes the residual urine acidity, like urine is acidic. So mucus protects sperm from the acid damage. So what is the erection physiology? Erection is mainly controlled by the parasympathetic system. So parasympathetic nervous system, which is S2 to S4 spinal segments. So what triggers an erection? Signals from sight, smell, touch, sound, imagination, and sexual thoughts. So these signals go to the hypothalamus, then to the spinal cord erection center. So what are the key events during the erection? So the first event is nitric oxide release. So in the nitric oxide release, the parasympathetic nerves releases the nitric oxide in the penis. So nitric oxide is a chemical messenger that causes the blood vessels to widen. And the second event is increase in CGMP. So there is no increase of CGMP inside the smooth muscles of the corpora cavernosa. So what does this do? CGMP makes the smooth muscles to relax. So these relaxed muscles, they allow more blood to fill the penile tissues. And the third event is smooth muscle relaxation. So as blood flows into corpora cavernosa, the penis becomes engorged. So that means the drained blood get compressed. So blood stays trapped. And the result is the erection. So here is a diagram which shows you the two systems. The central control, which is the brain and spinal cord control, also called as a peripheral control by S2 to S4 spinal segments. So in the central control, the hypothalamus, it interprets the sexual stimuli, dopamine and oxytocin, it supports the erection, and noradrenaline and serotonin, they reduce the erection, which is anti-erectile function. Whereas spinal cord control or peripheral control, the sacral erection center, it sends the parasympathetic signals to cavernous nerve. So this nerve, it releases the nitric oxide, which causes the erection. So this is the difference between the central control and peripheral control. So next is about the ejaculation physiology. So what is ejaculation physiology? Ejaculation has two phases again. The phase 1 is emission, phase 2 is expulsion. So phase 1 emission, this is controlled by the sympathetic nervous system T12 to L2 spinal nerve segments. So what happens in emission? The sperm plus fluids, they move into the posterior urethra. So this area, it acts like a holding chamber before the expulsion. So then contraction of the glands and ducts takes place. So vas differences, it pushes the sperm from testis, and seminal vesicles, they add the majority of the seminal fluid. Prostate, it adds the milky fluid like PSA, citrate and zinc enzymes. And then internal urethral sphincter closes, like this is located at the bladder of the neck. It prevents the retrograde ejaculation, nothing but semen entering the bladder. So why this is important? It prepares the semen for the next phase, like expulsion phase. So without proper emission, ejaculation cannot occur. Okay. So this is about the emission phase. And then after emission, there is an expulsion phase. So this expulsion phase, it is controlled by the somatic nerves, which are called as pudendal nerve, S2 to S4 spinal segments. So what happens in expulsion? Rhythmic contractions of bulbospongious muscle, pelvic floor muscles. So these contractions, they occur 0.8 seconds apart. And external urethral sphincter relaxes. So it allows the semen to pass out. And forceful expulsion of semen from penis. So they build the pressure, they create the pulsing sensation during the ejaculation, and they push the semen effectively out of the urethra. And here there's a diagram about the ejaculatory process. The first one is the brain. It inputs the visual, auditory, olfactory, touch, smell. So hypothalamus, it coordinates the sexual behavior. Dopamine increases, which promotes the ejaculation. And serotonin, the 5-HT1A, 5-HT2C, these can delay the ejaculation. And the second is spinal cord. So T12 to L2 spinal segments, which is the sympathetic center controlled by, and activates the emission process, which involves the vas differences, prostate and seminal vesicle organs. And the third is the sacral spinal cord, S2 to S4 spinal segments, which is controlled by the pudendal nerve. So this controls the bulbospongious muscle, ischiocavernosus muscles, and pelvic floor muscles. So this is responsible for the expulsion. And then last is the sensory feedback. So glans penis, it sends the sensory signals, and it helps to trigger the reflexive pelvic muscle contraction. So here is stage 1 and stage 2. Stage 1 emission is shown in this pink color image, and stage 2 ejection is showing you in the blue color image. So everything, the arrow which is colored in the pink color related to the emission phase, and the blue color related to the ejection phase. So in the emission, increase in the semen volume takes place, whereas in the ejection, semen is expelled. And the testosterone function. So what is the function and mechanism of the testosterone? So this is a main male sex hormone. It is produced by mainly Leydig cells in the testes. It controls male reproductive functions, the male body features, metabolism and behavior. So now how testosterone is produced? So the mechanism is shown in this picture. The hormone system works like a three-step chain of the command. So first you can see the hypothalamus brain. It releases the GnRH, which is a gonadotrophin releasing hormone. So this tells the pituitary it is start producing the reproductive hormones. So then anterior pituitary, it releases the LH and FSH hormones. And then testes. So this releases the Leydig cells which responds to LH hormone and Sertoli cells which responds to the FSH hormone. And it creates a negative feedback control system which involves high testosterone which tells hypothalamus and pituitary to reduce the GnRH, LH and FSH hormones. So this negative feedback, it also helps to maintain the stable hormone levels. So what are the functions of the testosterone? So testosterone, it acts as an testis, muscles, bones, hair follicles, brain and also the larynx. So these functions, they break into 4 easy categories like reproductive functions, secondary sexual characteristics, metabolic effects and behavioral effects. So in reproductive function, what is reproductive function? So this stimulates the spermatogenesis, the production of the sperms. So this works with FSH by acting on Sertoli cells. It also helps in formation of the male reproductive organs like penis, scrotum, testis, prostate and seminal vesicles. So what is the reproductive function? So it also increases the growth of penis, testis, scrotum during puberty. So low testosterone during childhood, it releases in delayed puberty. And what is the secondary sexual characteristics function? So testosterone, it causes the growth of the body hair, like in the places of face, chest, armpit and pubic. So it also causes the deepening of voice. So the larynx enlarges, vocal cords thicken. It increases the muscle mass, it increases the bone density and also the strength. And what are the metabolic effects? The protein synthesis increases. So this helps to muscle development. It helps in the faster tissue repair. And what happens when RBC production increases? It stimulates the bone marrow. It explains why men have higher hemoglobin levels than women. And fat deposition. So this shifts body composition toward more muscle and less fat. So fat deposition decreases. So low testosterone, this may cause the increased body fat, especially belly fat. So what are the behavioral effects? Libido and aggression. So testosterone affects the brain. Libido is nothing but it's a sex drive. And aggression and confidence, motivation and energy levels. So normal levels, it supports the healthy behavior, whereas very high levels, this may increase the irritability. So this is the function of the testosterone hormone. So next is male fertility. It depends upon the sperm count, sperm motility, normal morphology and also the function of axillary glands. So what happens when the andropause takes place? Andropause is nothing but changes in the aging. So after 40, what happens? There will be a gradual decrease in the testosterone. So it reduces the sex drive and obviously penis will have lower, penis will perform the erectile dysfunction and there will be a decreased muscle mass which reduces the sperm quality. Right. So after 40, men gradually decreases the sperm quality. So they do not have the much quality within the sperm before 40 and after 40. So this is the functions of the male reproductive system. So this is for today's session. In the next session, you will be going to learn about female reproductive system, anatomy and physiology. Thank you.